In this video, we are going to learn about the plasma membrane, cell membrane, chemical composition, molecular organization of plasma membrane, lipid bilayer model, trilaminar model of Danieli and Davison, Robertson's unit membrane concept, fluid mosaic model, and membrane proteins, along with conclusion and references. So first, when we just look at the basic idea about cytoplasmic structure. Cytoplasmic surface of all living cells is bounded by a limiting membrane called plasma membrane. It is elastic, porous and semi-permeable. Lipoprotein composition, semi-fluid consistency, laminar or micellar organization. There is an interface between exterior and interior of the cell and thereby regulates the exchange of materials between cell and its external medium. So, elastic, porous, semi-permeable, lipoprotein composition and laminar along with micellar organization. Laminar or micellar organization. Plasma membrane present in all types of living cells. In blue-green algae, bacteria, fungi and plants, a cell wall is present. In animal cells, cell wall is absent and in most cases there is only a cell coat or glycocalyx or cell lamina. When we just look at the cell membrane in detail, cell membrane is a biological membrane that separates and protects the interior of cells from the outside environment. Cell membrane consists of a lipid bilayer made up of two layers of phospholipids with cholesterol interspaced between them. Membrane also contains membrane proteins, including integral proteins that span the membrane and serve as membrane transporters, and peripheral proteins that loosely attach to the outer side of the cell membrane, acting as enzymes facilitate interactions with cell's environment. Membrane also contains membrane proteins, including integral proteins that span the membrane and serve as membrane transporters, and peripheral proteins that loosely attached to the outer side of the cell membrane, acting as enzymes facilitating interaction with the cell's environment. When you just look at the chemical composition formed of lipids, proteins, carbohydrates and water. Membrane lipids have a structural role. Permeability barrier include phospholipids, glycolipids, sphingolipids, phospholipids, plasmalogens, sterols. And you just look at phospholipids. Membrane phospholipids are the derivatives of glycerol, which is a 3-carbon alcohol. So they are called phosphoglycerides. A phospholipid molecule is formed of a glycerol molecule, 2 fatty acid molecule and a phosphorylated alcohol complex. Glycerol forms a backbone. Phosphate group complex forms the head of the phospholipid, polar and hydrophilic molecule, and the fatty acids forms its tail, nonpolar and hydrophobic. Now, plasmalogens, commonly found in nerve and muscle membranes. Phospholipid, fatty acid, saturated, unsaturated, and glycerol along with the phosphate. So, general structure of a phospholipid. A phospholipid has a similar structure to triglyceride. But with only 2 fatty acids and phosphate in addition. Glycolipids. Exclusively restricted to the non-cytoplasmic outer side of plasma membrane. Gangliosides are perhaps the most complex type of glycolipids. Sphingolipids are there, similar to glycolipids in structure, and the only difference is that in them glycerol is replaced by sphinginin as the core molecule. Sterols are nonpolar and absent in eukaryotes. Glycolipid is there, sugar residues there, lipid residues there, lipid membrane will be there. So membrane proteins, structural and functional molecules. Functional proteins serve as enzymes, permeases, receptor molecules and antigens, etc. Structurally of different kinds, globular proteins and alpha helix proteins. Membrane carbohydrates are of many types. Serve as cell coat or glycocalyx on the outer surface of plasma membrane. There are 2 categories of membrane carbohydrates, like complex polysaccharides and oligosaccharides. Molecular organization of plasma membrane. Plasma membrane has laminar or micellar organization. Lamellar hypothesis holds that the membrane is formed of molecular laminations, that is, sheet-like molecular layers of lipids and proteins. Micellar hypothesis holds that it is formed of globular subunits of lipoprotein. Laminar means sheet-like arrangement. Several models have been proposed to explain the molecular organization of plasma membrane. Laminar and globular. These are the major one. Then fall under two categories, lamellar and micellar. Membrane models is of two types, like lamellar models are there, micellar models are also there. So lipid bilayer model, trilaminar sandwich model, Danieli-Davison model, unit membrane model, crater membrane model, lipid pillar model, and Benson's model, Lennard-Singer model, mosaic membrane model, and fluid mosaic model. Lipid bilayer model or Gorter and lipid bilayer model of Gorter and Grendel. In 1926, Gorter and Grendel postulated the lipid bilayer hypothesis. According to it, cells are covered all around by a 2 molecules thick lipid layer. According to it, cells are covered all around by a 2 molecules thick lipid layer. In this bilayer, the polar ends of the molecules are directed outward and the non-polar ends inward. So extracellular region is there, intracellular portion is there. Phospholipid bilayer can be seen, hydrophobic tail into the inside, then hydrophilic head into the outside. Trilaminar model of Danieli and Davison, that is also called as a sandwich model, proposed by James Danieli and Davison. According to this concept, plasma membrane is the 3-layered structure. It consists of a central phospholipid bilayer set between 2 peripheral protein monolayers such as butter or egg in a sandwich. The lipid molecules of one layer maintain linked with those of the other layer by their non-polar tails through van der Waals forces. They link with proteins by polar ends through ionic linkages, hydrogen bonds, or electrostatic forces. Danieli and Davison model. Proteins form distinct layers, sandwich, globular protein on the top, hydrophobic zone and hydrophilic zone after that. So protein coat will be there, lipid bilayer and Danieli and Davison model, and in between them polar pore will be there. Next is the Robertson's unit membrane concept. David Robertson proposed the unit membrane concept. The model proposed by him is mostly based on the Danieli and Davison trilaminar concept. Robertson suggested that all biological membranes have the same trilaminar structure with approximately 75 angstrom thickness. So to such a universal membrane, he gave the name unit membrane. Central zone and marginal zone as there. Central zone is a lipid bilayer described by Robertson as the lipid bilayer leaflet. Marginal leaflets are thin and flat protein monolayers. Major points of Robertson's views are the following. Any piece of biomembrane comes from a pre-existing membrane. Marginal layers are thin and flat protein monolayers. Major points of Robertson's views are the following. Any piece of biomembrane comes from a pre-existing membrane. In other words, membranes make membranes. Membranes of ER are extensions of plasma membrane, and so the space inside them is a part of extracellular space. Differs from Danieli-Davison concept by membrane proteins have definite structure and functional roles. The arrangement of the membrane molecule is asymmetrical. Inner surface coated with simple proteins and outer surface with mucoproteins and mucopolysaccharides. It upholds 3 concepts. The central lipid core of the membrane is structurally and functionally important. Proteins do not have significant structural and functional role. Proteins and lipids have perfect symmetrical and continuous arrangement. Even though basic ideas of the model are still valid, their trilaminar model seems to be an oversimplified scheme. In this, proteins are entirely outdyed lipid bilayer. Studies have shown that lipids and proteins are asymmetrical in arrangement. Extracellular medium is there, outer protein layer is there, hydrophilic heads of lipids, hydrophobic tails of lipids, intracellular cytosol, inner protein layer, unit membrane model of plasma membrane. Why it is called unit membrane? All biological membranes have the same trilaminar structure with approximately 0.5 angstrom thickness. That's why it's termed unit membrane. Shortcomings. Which are the shortcomings of unit membrane? Unit membrane is an oversimplified version. It does not satisfactorily explain the permeability and transport properties of the membrane. It simply regards the membrane as a rigid and inert structure. It does not account for the dynamic nature of the membrane and also for the molecular mobility of its protein and lipid constituents. It simply considers the membrane as a static structure. Next is the fluid mosaic model. Fluid mosaic model proposed by Singer and Nicholson in 1972. According to it, plasma membrane is a two-dimensional quasi-fluid solution of organized lipids and globular proteins. Plasma membrane is a bimolecular quasi-fluid lipid structure in which proteins freely float, move about and interact. There are major features like plasma membrane is a lipid dynamic structure existing in a liquid crystalline or quasi-fluid state with mosaic molecular arrangement and free molecular movement. Fluid mosaic model can be seen. Extracellular fluid, carbohydrate is there, transmembrane protein and glycolipid along with glycoprotein is there. So cholesterol can be seen and peripheral protein will be there. The fluidity of the membrane is directly proportional to the chain length and unsaturation of its fatty acids. The major components of the membrane are held together through non-covalent interactions. The cohesive forces between lipids and also between lipids and proteins are weak hydrophobic interactions, ionic and hydrogen bonds. Membrane lipids and proteins undergo free lateral diffusion. Vertical diffusion of proteins and lipids is very much restricted. Membrane phospholipids are arranged in a bilayer that serves as the structural framework, as a permeability barrier and also as a solvent for integral membrane proteins. Membrane lipids interact with proteins for the stabilization and proper organization and functioning of membrane. According to the Singer-Nicholson concept, phospholipid molecules have a continuous arrangement. They are arranged side by side and back to back in parallel rows, forming a continuous lipid bilayer. This lipid bilayer forms the structural framework of the membrane. Its outer monolayer is formed mostly of acidic phospholipids and the innerons consist mostly of neural phospholipids. Their polar heads are directed outward of the aqueous phase. Non-polar fatty acid tails are directed inward to form the non-aqueous central core. This arrangement enables maximum interaction between polar heads and water molecules. The two lipid monolayers remain linked with each other by their two fatty acids, tails to hydrophobic interaction forces. The membrane phospholipid layers are in a mobile fluid state, free to diffuse laterally within the molecule. The phospholipid bilayer is essentially a film of oil and it makes the membrane semi-fluid. Cholesterol occurs tightly bound in the phospholipid. We can see the structure. Extracellular fluid is there, fluid channel transport protein, globular protein is there, glycoprotein is there, carbohydrate is seen, hydrophilic heads and phospholipid bilayer, phospholipid molecule is there along with hydrophobic tails, alpha helix protein, integral protein, surface protein, filaments of cytoskeleton, integral protein like globular protein, peripheral protein, cholesterol and glycolipid. This is the structure of membrane. Membrane proteins. Membrane proteins are mostly globular without orderly arrangement. They are randomly dispersed and discontinuous. They remain partially or completely inside or outside the lipid bilayer. The proteins completely outside the lipid bilayer is called extrinsic or peripheral protein. Those which penetrate the lipid bilayer is called integral protein. Integral protein amount to 70 to 80% of membrane protein. Some integral proteins span the entire lipid bilayer. They are called transmembrane proteins. They form pores through the membrane and also transmit signal from one surface to membrane to the other. Intrinsic protein usually float and freely move about within the lipid matrix just as icebergs float and drift in the open sea. The dispersed distribution of proteins forms the mosaic part of the fluid mosaic model. There are extrinsic proteins which are hydrophilic. They bind with the membrane surface by electrostatic interaction and hydrogen bonding. Most intrinsic proteins are amphipathic. Here we can see a structure. Integral protein is there, cholesterol is there, globular protein is there, glycoprotein is there, carbohydrate will be there, so glycolipid will be there. peripheral protein, alpha helix protein, channel protein will be there. The hydrophobic portion resides within the lipid bilayer. Hydrophilic part projects out of the lipid bilayer. In fluid mosaic model, carbohydrates are entirely outside the lipid bilayer. They bind with lipids and proteins, forming glycolipids and glycoproteins.